We now switch from stem cells to gene therapy, and it's a pleasure for me to introduce Matt Porteus. Matt is an associate professor in the Department of Pediatrics, and also is an active physician scientist working in the division of stem cell transplantation and regenerative medicine that I have the privilege to lead. So Matt really was the first one to demonstrate that human cells can be indeed corrected in gene editing, and I let him to tell you what is next.
Okay, thank you very much. Thank you for the introduction. Thank you for the organizers for inviting me. So I'm going to give a slightly different talk than others in the sense that what I want to emphasize is the program that we want to develop here at Stanford that uses correcting mutations to cure childhood diseases. And as the title of my talk says, it's not science fiction anymore. We can actually do this. So I'm surprised that in this Childx conference, I'm the first person that gets to show a lot of pictures of random cute babies. So as many of you know, and as Dr. Guttmacher mentioned in his talk, there's 4 million babies that are born every year in the U.S. And all of these babies at birth have a little bit of blood squeezed from their heels and create the Guthrie card. And depending on what state you're in, this Guthrie card gets analyzed for a number of different inherited genetic diseases. In California, that list of diseases is over 50. But again, as was mentioned earlier today, is soon what will basically happen from the spot of blood is the entire genome of everyone is going to be sequenced at birth, and the identification of genetic predispositions or overt genetic diseases will be done at birth by sequencing. Interestingly, a couple weeks ago, a paper came out of Iceland where they sequenced the genomes of 2,000 quote, healthy individuals, and it was found that somewhere around 7% of those individuals have evidence of actually having mutations in 2 copies of the same gene, suggesting that 7% of us may have actually a genetic disease. So these genetic diseases, as again, Dr. Roncarolo mentioned earlier, and as well as Dr. Andrews, are for the most part quite rare, but since there's so many of them, they actually really permeate our world of pediatrics. And when I don't attend on the hematology service anymore, but when I did attend on the pediatric hematology service, it was frequent that every patient on the service had a genetic disease, whether it was sickle cell disease, thalassemia, hemophilia, or some variant, some of the other genetic diseases of the blood. But essentially these diseases encompass all of the subspecialties that we deal with. And depending on who I'm talking to, I can add and subtract various different diseases from this list. And as has been mentioned, we estimate now there's about 10,000 of these diseases, but I think certainly in several years we're going to say that there's even more of these diseases. I'm going to focus on 2 specific diseases, sickle cell disease and severe combined immunodeficiency. So on the top, sickle cell disease is caused, its name is given because you can see the red blood cell adopts this abnormal sickle cell shape, and it's caused by a single mutation in codon 6 of the beta globin gene. And what you can see is there's a little red T instead of an A. So a single adenine gets converted to thymidine in this gene, and when you have 2 copies of that change, that leads to the disease. Severe combined immunodeficiency, otherwise known as bubble boy disease, shown in the lower panel, the mutations are caused in a gene called the interleukin-2 receptor common gamma chain gene, which is a mouthful, and the mutations span the entirety of the gene. But nonetheless, any given patient only has a single mutation. What's interesting about this is that in the 6 billion character genome that we have, which would be equivalent to a 1.1 million page book, which I guess could be fit on my iPad, there's a single typographical error that causes the disease. And what we've been working on, and now a number of other people have been working on, is the idea of using genome editing to correct that typographical error. And that sort of, again, 10, 15 years ago when we started working on this, that sort of sounded fanciful and out of reach. But I can tell you now that we can do that. So how do we do genome editing? Well, the way we do that is we engineer specialized proteins that are designed to recognize the DNA sequence around the mutation. And now there are a number of different classes of proteins called ZFNs, talons, homing endonucleases, CRISPR-Cas9, all of whom fundamentally act through the same way, which is they are designed to recognize a specific site in the genome. They create a double-stranded break in that genome, depicted here as splitting that blue bar into two. And what we then do is we simultaneously provide another piece of DNA that has identical to the site that we've made the break in, except it no longer has the mutation. And the cell, using a process called homologous recombination, will integrate that donor correction DNA and correct the mutation. It's actually functionally a copy and paste mechanism, so an Apple C, Apple V sort of approach that we're taking advantage of here. And so what we end up with is a cell with the mutation in the gene corrected. And this process gives us nucleotide resolution and is true precision therapy in terms of we know exactly what we're doing to the genome. What was also mentioned earlier was the problem of not every protein is druggable, but because our nucleases can be designed to recognize essentially every site in the genome, we can target every single site in the genome. We can correct every gene, we could correct any genetic defect. So fundamentally, this approach allows us to say nothing is undruggable. So it's schematized, we have a cell, we introduce our correction fragment, and voila, we correct the gene. And the strategy is, as much as Dr. Andrews described, is what we would like to do is identify a patient at birth, perhaps through newborn screening, or perhaps even better, which is prenatally through genetic diagnosis, harvest the hematopoietic stem cells from that patient, use our gene correction strategy ex vivo, correct the gene, take those corrected cells, and transplant them back into the patient. So if we could do that, then this idea, because we have precision therapy, then could be expanded into a number of different axes. So the obvious axis is that you could say, well, if you're correcting sickle cell disease or SCID, we could start using this approach to correct other genetic diseases, such as hemophilia or cystic fibrosis, or the whole list of 10,000 diseases. But the ability to precisely modify the genome of a cell also allows us to think about engineering cells to secrete therapeutic proteins. And those therapeutic proteins might be useful in treating rare but currently uncurable diseases, such as congenital heart disease or type 1 diabetes. They may also be able, you could use a cell-based therapy that's delivering a protein therapeutic that might cure common but currently incurable diseases, such as cancer, neurodegenerative diseases, allergy, and HIV. So what are we going to need to do to achieve this here at Stanford? Well, it's going to take a vibrant, collaborative, and an interactive community to succeed. We need to have the technical expertise in both stem cell biology, transplantation medicine, genome editing to occur, and that is, I'm wonderfully situated in the Loki Stem Cell Building to do this. We need the support of the university, we need the support of the Stanford School of Medicine, we need the support of the Lucile Packard Children's Hospital, and we need the support of our local community, shown in the lower right-hand corner. And all of that is what's going to need to be done to really translate this process. We're going to need support to convert what we do in the laboratory to a clinical scale process. So in my laboratory, we can take about 1 million cells and we can deliver our nucleases and our donor vector, and we can get correction on the order of 1 to 5%, which is actually clinically reasonable. But what we're going to have to do clinically is we're going to have to scale that up by 100 to 200 fold. And so you can sort of see that process. And that's not an easy process. So, but we can do it. The other thing we need is a specialized facility, a specialized manufacturing facility that will allow us to engineer these cells such that they're safe to give to patients. And Stanford has now committed to building this facility, and it's going to be online in January of 2016. And we need to recruit someone skilled to run this facility. And again, we're very fortunate to have recruited Dr. DeGiusto from City of Hope to run this facility, a tremendous experience. Ultimately, what we need to do is not clone Dr. DeGiusto. I think that would be a bit much, but we're going to need to build bigger facilities and recruit more people, because the idea is not necessarily just treat 1, 2, 3, or 4 patients, but to treat tens, hundreds, and thousands of patients. We need the support of Lucile Packard Children's Hospital, because these first patients are going to need to be specialized housed. We're going to need to have the specialized infrastructure. And again, the extension of the new hospital provides us with that infrastructure infrastructure. And ultimately, we want to have partnerships with industry and biotech, because as much as we're proud about bringing these novel curative therapies to Stanford and delivering them at Lucile Packard Children's Hospital, ultimately these patients live around the world, and we want to be able to deliver these innovative therapies around the world. And the best way of doing so is partnering with companies that have the infrastructure, resources, and expertise in terms of doing this. I will say there's always going to be a role for academics in this process. First of all, biotech and pharma, for very good reasons, is less prone to take on high-risk proof-of-concept curative studies, and that's where we fit in. Second of all, while Dr. Andrews suggested that GSK is willing to take on ultra-rare diseases, we know that there's going to be N of one, N of few diseases. And again, I think academics plays an important role in developing curative therapies for these ultra-rare diseases. And then finally, I think since we're a nonprofit and we come from perhaps a more idealistic setting, we can be advocates to make sure that these curative therapies don't just get to our local communities, but get to the parts of the world that are resource poor. And to end, I would just like to, end with the final quote, which I think hopefully encompasses a lot of what's going on in terms of improving child health, which is a quote from Kurt Vonnegut, who said, "I want to stay as close to the edge as I can without going over. Out on the edge, you can see all kinds of things you can't see from the center." So thank you for your attention, and I look forward to taking questions when we have our panel discussion.